Okay, good morning, ladies and gentlemen. Welcome to Elixir Friday Coffee. Today, Ondřej Vávra from Loschmidt Laboratories from Masaryk University will tell us about CaverDock and how it can be used for analysis of ligand transport through the protein tunnels. Ondro, the floor is yours.
Okay. Good morning, everyone. Thank you very much for the introduction. Yeah, as was already mentioned, I'm going to talk about CaverDock, which was developed at our lab together with Dr. Jiří Filipovič from the Faculty of Informatics. But before I start talking about CaverDock, I would like to mention a few things about why we decided to develop this tool. So first of all, in our laboratory, we focus on the study of enzymes, which are proteins which are capable of catalyzing chemical reactions. High percentage of these enzymes have a buried active site somewhere inside of their structure. And then this buried active site is connected with the outside environment by access tunnels. And these tunnels are then used by the substrates, the molecules that go into the protein to take part in these chemical reactions and products of these reactions to get out. The study of these access tunnels is important not only in basic research, but also, for example, for very important in different types of industries, for example, the production of drugs and pharmaceutics in pharmaceutical industry or in food industry, and etc., etc. So how can we study the ligand passage in proteins? Of course, the most used or the original approach would be to study this experimentally, but unfortunately, experimental approaches for this kind of studies are very financially expensive. You need a lot of equipment and trained specialists to do these kinds of studies. But of course, on the other hand, we have theoretical approaches or computational approaches. On one hand, we have molecular dynamic simulations, which use a high degree of theory, but are computational and time demanding. And on the other hand, we have geometrical methods, which are unfortunately too approximative, but at least they are fast, but will not give us any information about the energy of these binding or unbinding processes. So that's why we decided to develop CaverDock, because so that it would be fast and easy to use like the geometrical methods, but also we wanted to incorporate some more advanced features so it could also describe the energy of these processes. So what CaverDock is using is iterative molecular docking. Just to mention here, molecular docking is a method in molecular modeling where you have your protein and you analyze its binding site and you simulate calculations where you try to find out how certain ligand or molecule would fit in this active site of the protein, or also you can use, for example, larger set of these molecules and also to discard the molecules that do not bind properly or should not bind. Specifically, CaverDock uses optimized algorithm from AutoDock Vina for docking, and in our case, we use it iteratively. So not only that we analyze the active site itself, but also each part of the access tunnel. And CaverDock also allows ligand flexibility and sidechain flexibility when it's turned on. So how does the CaverDock workflow look like? So first you start by picking the protein you want to study, and then you need to calculate the tunnels using tools such as Caver. Then you need to extract the 3D representation of this tunnel and discretize it into a set of disks using our discretization tool. This is a very important step because each disk represents one step in the simulation, and it is used to guide the ligand through the tunnel so it can get out of the protein or into the protein. And once you have your protein, your discretized tunnel, and the ligand, you can then run the CaverDock simulation. Here I prepared a short video showing how it looks like. So we have the ligand traveling through the tunnel all the way to the binding site of the protein. You can see how it moves and gets all the way in. As I previously mentioned, the great benefit of CaverDock is to calculate the binding energy and being able to describe the binding process energetically. So in comparison with regular molecular docking approaches, these would this would focus on the best binding site, for example, in this case represented by the green point and also the green structure of the molecule inside of the tunnel. But since we also analyze the whole tunnel, we can actually find places which are more difficult for the ligand to pass through. For example, the case of the red point in the energy profile and also the red structure of the ligand in the tunnel, where the ligand is actually more narrow and it's difficult for the ligand to pass through. So these places then could be, for example, optimized by mutagenesis of the protein to improve the properties of the protein, so even larger ligands could fit through. Something like that was studied in one of our case studies, where we followed previously published molecular dynamic simulations, and we repeated the experiments with CaverDock. We analyzed tunnels in haloalkane dehydrogenase variants, specifically LIN-B variant, and we simulated the unbinding of 2-bromoethanol. If we look at the studied variants of the LIN-B dehydrogenase, we looked at the LIN-B wild type, the variant 32 and 86. Specifically, we studied the P3 tunnel. In the pictures, it is shown as the red tunnel going to the back of the protein. You can also see that in the wild type and the 32 variant, it's quite narrow and closed, while in the 86 variant, it's much more open. When we ran the CaverDock simulations, we saw that it is in perfect agreement with the tunnel analysis and also the previously published MD simulations, that in the case of the LIN-B 86, the product molecule is able to leave through this P3 tunnel, and it has much higher difficulty to pass through in the wild type and the 32. In a different study, we focused on screening of larger pool of molecules. In this case, we analyzed leukotriene A4, which is a human enzyme, and in our bodies, its main function is the biosynthesis of pro-inflammatory mediator. In this case, we analyzed two different tunnels going from the active site to the surface of the protein, and we used screening library of 56 anti-inflammatory drugs. For the results, from each energy profile for every molecule, we extracted the barrier, the highest point in the profile, and then we compare the distribution of these profiles for all the 56 molecules. And in this case, we got a really nice result that showed that the PGP tunnel, the red one, would probably be more biologically relevant and probably more used by these molecules and drugs to get in and out of the protein rather than the LTA4 tunnel, the blue one. Now I would like to mention several projects that were based on the development of CaverDock and are now used as extensions of this tool. So the first one is CaverWeb, which is our web interface for the tunnel analysis and the ligand passage analysis by Caver and CaverDock. This website is aimed at less experienced users, so they can do all these analyses on the web and they don't need to install any software on their local computers. We prepare very clear and easy to use user interface. So in order to get results, it's very simple in this case. Then with colleagues from Visit Lab from the Faculty of Informatics, we developed DocVis, which is a tool for the analysis, detailed analysis of results from CaverDock. It's using modules which were previously developed for a different tool called Caver Analyst. And it's completely interactive and synchronized between all the four windows that are in the user interface. So on the top right, you have the part where you can analyze energetic profiles of the transport. You can simultaneously analyze several ligands and also the properties of the tunnel. In the bottom right part, we have a module that shows the tunnel lining residues and their physical-chemical features. So you can look at what kinds of residues there were and how much impact they had along the tunnel. And of course, on the top left, we have the 3D visualization, and in the bottom left, we have a cut through the tunnel, where not only you can see the tunnel lining residues at specific disk of the discretized tunnel, but also the interactions of these residues with the studied molecules. The last project where we also use CaverDock is the CaverDock API, which was co-developed with Martin Shukrovsky and Jakub Beránek from IT for Innovations. Here we re-implemented CaverDock parts as modules and objects. And in this case, it's aimed at advanced users of CaverDock, so they can easily do much more complicated studies like batch analysis and so on. So using this API, they can then automatize the whole pipeline and also easily evaluate the CaverDock jobs. I'm now getting at the end of the presentation. So in conclusion, I would like you to remember that CaverDock is a new, fast, and easy to set up method for the study of ligand binding and unbinding through protein tunnels, and also that you can use it on different types of studies. Either you can study whether certain ligand can pass through a tunnel of a protein, or compare different ligands or different tunnels or different protein variants. Here I would like to say thank you to everybody in the lab who took part in the development, and also to all the financing agencies. And thank you very much for your attention. If you'll have any questions, I will be very glad to answer them. Thank you.
Thank you for a nice lecture. And now there is time for questions. So I see 2 questions so far. First is from me, so I will start. My first question is, what physical chemical features DockViz is showing?
Yeah, so first of all, of course, the charge. Also, then you can switch it to hydrophobicity. And then we have several more which are the same as you can find them in Caver Analyst. Unfortunately, at this moment, I do not remember the complete list. I'm sorry.
Thank you. And second question is from Marianne. How often is the nearest point in tunnel also the site with the highest energy? So basically,
I think that, yeah. Actually, it was, for example, in this case of the protein I showed there, that's actually a real energy profile. But usually it happens somewhere in the middle rather than close to the surface. Yeah, from what I remember, we analyzed so many proteins, and usually the opening is quite wide, but then the bottleneck appears somewhere along the tunnel rather than at the beginning.
And yet here are the highest energy at the opening.
Yeah, but this was selected as a nice demonstration that you can find this place somewhere along the tunnel at the start. This is mostly used as a demonstration that it looks really nice. Yeah, but it's correct that in this case, the narrowest bottleneck was actually at the beginning of the tunnel.
So my next question is, how many protein ligand pairs have you validated?
Well, in the published validations, we studied maybe around 30 tunnels altogether in the studies we published. That would be also, when it comes to the ligands, around hundreds, let's say. Also, we use much more in the testing of CaverDock. And right now I'm working on a very large project where we're using it to annotate a certain database of proteins, where it's around 30,000 protein ligand pairs.
Cool, thank you.
Yes.
So, any other questions? Okay. I don't see any more, so that's it. So thank you again, and for the really nice lecture, and see you next week on PrankWeb.
Okay.
And thank Ondřej Vávra again for his lecture.
Thank you. Have a nice day.
Bye. Have a nice day.